Hello, this is Tova Bergston, an MD PhD student at the University of Illinois at Chicago College of Medicine. This video is part of our Survivors Advising Scientists educational program, a bidirectional educational platform meant to connect our communities and increase scientific literacy and engagement. Our goal is to bring everyone to the table for scientific discussions related to cancer science and therapies. This is video number 8, Study Bias and Randomization. The first thing we're going to cover is bias. Bias is an adjustment in the design and/or conduct of a study. It may be intentional or unintentional, but in any case, bias can affect the results of a study, which is why it's important for researchers to be aware of and try to avoid bias. There are many types of bias, which we'll go through now. Selection bias is when researchers select participants for their studies that do not reflect the population as a whole. For example, if researchers only enroll Caucasian participants into a clinical trial when the actual population that would receive this treatment is more diverse. As you might expect, this can affect how one interprets the results of a study. So, if the drug being tested in this clinical trial is determined to be effective, researchers can actually only say that it is effective for the patients who match the population that they studied. In our example here, this drug could only be considered effective for Caucasian patients, but not the more diverse general population. Another type of bias you might encounter is called measurement bias. This happens when the dependent variable, or the outcome measurements that are made by scientists, do not properly address the research question. For example, if researchers are trying to study tumor spread, but they only measure the size of a primary tumor, the measure they are using is not addressing the research question. This can affect how one is able to interpret the results of the study. If researchers are measuring the size of a primary tumor, which has no relation to tumor spread, so any claims made about this study in terms of tumor spread would not be valid until a measurement is used that actually studies tumor spread. Expectancy bias is when researchers' own beliefs affect the experimental results. For example, if an inventor of a drug is so confident in their creation, they are overconfident it will succeed and therefore might interpret the results of an experiment in their favor to match what they expect to happen. This affects how one can interpret the results of these studies. So, if this inventor was so confident in the success of their drug, he or she may interpret the results in a more positive way in order to claim the drug is successful, when in reality it may not be. Observation bias occurs when a researcher's recall memory affects the interpretation of results. For example, if researchers are so accustomed to certain medical readings that they expect to see results similar to what is typical according to their memory. This can affect how one would interpret the results of a study. If a researcher is accustomed to a test value being one number, here 120, based on years and years of practice, they may expect results similar to this number and may not pay careful attention when recording test results, which incorrectly changes the study's outcome. Reporting bias occurs when researchers only report a select sample of the results. For example, they may only publish what they want the public to see, or they may only publish positive results. This can affect the results of a study. If a study has 3 outcome measurements and only one was considered successful, and researchers only report that one outcome, it makes the experimental treatment seem very successful, when in reality it failed at 2 outcomes. Next, we'll discuss blinded experiments. Whether on purpose or not, biases, which we've discussed in the last several slides, can affect experimental results. Researchers use something called blinding to prevent biases like these from affecting their studies. For example, if a patient knows he or she is receiving a powerful drug, he or she may change their behavior to be more positive, like, I'm feeling good about my treatment, so I'm going to keep getting healthier. The solution to this is to use single blinding to keep patients from knowing which treatment group they are in so they don't change their behavior. This will help make sure the results of the study are actually due to the treatment and not a change in patient behavior. In another example, if a doctor knows which patient is getting which type of treatment, they might change their behavior in order to help their favorite treatment succeed. For example, I know this patient is getting this medication, which I really like, so I'm going to spend extra time with these patients and make sure they're doing great. The solution to this is to use double blinding to keep both physicians and patients from knowing which treatment group patients are in, so neither party changes their behavior. This will help make sure the results of the study are actually due to the treatment and not a change in physician or patient behavior. Usually, double blinding requires a third party who keeps track of which patients are in which group and receiving which type of treatment. Researchers also use something called randomization to prevent confounding factors, which are another type of bias. If you'd like to know more about confounding factors, please review Module 4 of this program. Randomization is the best way to keep experimental and control groups as similar as possible. Here we have two examples. On the left, a randomized experiment shown in green, where the groups have similar gender ratios. The experimental group is composed of 51% female and 49% male patients, while the control group has similar percentages, with 49% female and 51% male patients. This means that gender will not bias the outcome of these results, thanks to well-done randomization. On the right, in red, you can see a non-randomized experiment. Here, the experimental group only has 29% female patients, but the control group has 71% female patients. Because the experimental and control groups are not equal in terms of their percentage of male and female patients, the results of this study may be biased by gender, so the results are then affected by a lack of randomization. Now, we'd like to go through some questions that you can think of when you read the results for a clinical trial. They may help you determine how you would feel about the results. Was the study randomized? Was the study blinded? What type of bias might be affecting this study? For example, were only certain populations of patients included in this study? How might the design of this study affect the results? This is the end of Module 8. Now, the presenters would like to thank Dr. Kitievsky and the administrative staff of the University of Illinois Cancer Center for their support in this effort. Additionally, we'd like to give a special thank you to all the cancer patient advocates who provided feedback during the development of these modules. We'd also like to thank you for reviewing this module, and we invite you to send any feedback on this module or suggestions for future content. Please feel free to move on to the next module, number 9, on how to read a scientific poster. Thanks again.